discussing an automatic pipeline that annotates gene expression image sequences using two models, a nonparametric factor analysis and conditional random fields. As the previous two speakers already brought this in, nowadays we are dealing with large amounts of high-dimensional data sets, and obviously this field is developing, and I'm quite excited to be part of it. And so throughout this project we had three goals in mind. The first one was to look for similarities and patterns inside these such massive and complex data sets. Also, we are always thinking how to model time-dependent data. We are dealing with either Drosophila embryos or cancer progression data, and we were aware of time dependency, and we are looking for such modeling. And also, as we are dealing with real data, we are trying to analyze, some scenarios where data would be missing, and how can we address that? And so these are the goals, and I will try to give an answer to each of those at my best. Going back a little bit to high-dimensional data, we used a similar protocol that has been introduced by the previous speaker, to which I would like to thank. We used RNA in situ hybridization. This time we had images from Drosophila embryos. And I'll get into more details about acquiring those images, but I would like to point out with this slide the complexity of our data. So we had roughly 100,000 images, and as you can see here, all these embryos are at different moments in time, different moments in their development, and there will be images that will show complete or partial embryos, like maybe you can see maybe here like a partial embryo. There will be images that will show a bunch of embryos at the same time, and so of course the analysis will just get more complicated, not to mention the images that will be in or out of focus. And so just so you have an idea, we will have over 100,000 images, and many of them will be not in the ideal case. The second point I mentioned initially was the temporal analysis, and we are aware from the very beginning that we will have at one point to deal with this transition that happens across stages throughout Drosophila embryogenesis. And I'm going to give you an example here. We're showing two genes, CYP1 and EVE, and we show embryos that are going from stage range 1-3 all the way to stage range 13-16, and you can clearly see how the gene being expressed throughout these dark blue areas, there is a dependency with time, right? And of course we would like to model that as well. I'll give you all the details in the following slides. And so let's get back to the outline of my talk. The main goal, as I mentioned initially, was to come up with a pipeline that would automatically annotate Drosophila embryos using a fixed vocabulary, a fly-based vocabulary of words. And to do that, we used two different models. The first one was used to do feature extraction. So I developed this sparse Bayesian factor analysis model, and pretty much I answered the question of how to represent Drosophila gene expression patterns in a meaningful way, and I will get to that shortly. And the second part was to address the temporal data, and how do we model temporal data, and how do we actually manage missing data. And the answer, of course, was using conditional random fields. At the end, if you're still with me, I would like to talk a little bit with you about nonparametric Bayesian models and a few conclusions. Going back to our data, as I mentioned, we used an RNA in situ hybridization protocol. This entire data set was collected, and it's still collected in San Francisco at Lawrence Berkeley National Laboratories. And the project started about 12 years ago, where the researchers were interested in analyzing the spatial and temporal dynamics of gene expression in Drosophila embryos. So they've decided to start taking pictures of various embryos in which given a gene is being expressed. So you see here lots of embryos, and they will actually be fixed and in this 96 well plate protocol, and images will be taken of various embryos across time throughout Drosophila embryogenesis. And you see like a snapshot of one of the wells used in this plate. And up to date, there are over 110,000 images recorded of these embryos, and they cover roughly half of the genome, about 7,500 genes. And their goal was, of course, to study the dynamics, the spatial and temporal dynamics of gene expression, and they had a specialist that would manually annotate each and every image with a fixed vocabulary. So that's where I came in place, and I've decided to run something interesting to actually make this process automatic, to save that guy probably years of work. A little bit more about Drosophila embryogenesis, just so you get a better understanding of the biology behind the project. Drosophila embryogenesis spans over 20 hours or so. It starts right at the pronuclear fusion, and it goes through pretty much 17 stages, biologically defined. But for us, we've decided to group the first 3 stages and the next 3 and so on into what we call stage ranges, because not so many biological differences happen between, let's say, these 3 stages. So from our analysis, we just decided to group them together to ease our computation. And so the specialists at Lawrence Berkeley Laboratory would take images, we take pictures of embryos for every single stage range throughout development, and for all these 7,000 genes. All right. The first step that everybody would have in mind when we'd have to deal with hundreds and thousands of images would be how to do image registration. And I was lucky enough that actually there was already published some work in 2010 in bioinformatics that would actually do this very thing. And by that I mean to take every single real image that might look something like this, and to isolate every single embryo from the image, the embryo that is in a full shape, and then rotate it into a standard orientation. And of course, we really wanted to do this so that we can start our analysis. And just to have an idea, after we apply this already, the already published framework, we ended up with about 52,000 to 53,000 images out of the the 100,000. Many of them just didn't make it through the pipeline because either quality controls or they were out of focus. And so the coverage would be like an early stage 4-6. You'd end up with maybe 3,000 genes, recorded over 9,000 images. Later on, as time progresses, more organs, more biological processes are taking place, and so for pretty much the same gene count, you end up with more images just because they are more interested, and so the specialists will take more images, will acquire more images. Besides those 50 and so thousand images, we had to select these annotation terms to run our annotation. As I initially mentioned, there were about 500 annotation terms in Flybase in this vocabulary, but we didn't really want to run our pipeline through every single annotation term, so we looked only across the most used annotation terms, and we selected 14 most used annotation terms for stage range 4-6, and as an example 8 for the later stage, just so we would cover about 85% of the genes that we're looking for. And we also looked only across the genes in common of all these 5 stages, which covered about 1,700 genes. And you have the numbers, the new image count here. Now, this is just one last example before I go to the actual analysis. I'm showing one gene and I'm showing one gene and then the registered images. This is one image from stage 4-6, and you can see later specialists would have to acquire maybe 12, 15 images because more processes are happening, and they would like to to analyze those in more details. And also, you can see some annotation terms that would actually annotate these very images. And in bold, I'm showing some of the important terms that a manual curator will actually annotate with a given image, and this is what we actually wanted to do automatically. We would like, if I would get a new image, once I have my model trained, I would like to automatically annotate from this set of terms. I'd like to tell you which one would actually annotate that given image. Now, in terms of feature selection, I've already heard in the previous talk a very interesting idea would be to use a bottom-up approach. You can, run given an image. You can run some PCA. You can do wavelets. You can do SIFT. You can even do some virtual representation of those images, and you end up with a rather large set of features in the thousands. And one problem that might come here will be the redundancy across your features. If you really want to run this, in a pipeline, if you want to make this really feasible, you would really like to minimize the number of features that you're extracting for a given image. And what sometimes people do, they look at a top-down approach, which would finally generate a much smaller number of interpretable features. And this is what I did. And I wrote a sparse Bayesian factor analysis model that would extract features for every single image, but would start looking at the top, like over all the images, and then would extract features separately, and you would end up with a much smaller number of interpretable features. And of course, we compared our results with the state-of-the-art results that used bottom-up approaches, and we showed that we did at least as good, even better. A few details about the sparse Bayesian factor analysis, the feature extraction. X is our data. You look here, X would be our entire image collection. Every single row here would be represented by one image, and the factor analysis would decompose our original image set into two matrices. Like in a mixture model, you will have a matrix of loadings A and a matrix of factors S, which you can think of as mixtures. And both matrices A and S will be inferred by the model with no intervention from the user, and as well you would need some error matrix to compensate from the differences between X and the product A times S. And just so you better understand this, I just show this picture here where X would represent your entire set of images. Your matrix A will contain the loadings in your model, and your matrix S would contain the factors. And once we run this throughout all of our images, we're going to look at every single row in matrix A, and those will be our new features that we're going to use for further analysis. Now, as you can see, there is a number K here. This dimension that has nothing to do with the original image set is not P nor N related to X. And this is actually the number of factors that we had to select in our model. When we published this work in 2011, we ran multiple cases. We set this number anywhere between 60 to 150, and we decided which would be the optimal number of factors. But since then, we actually had another idea. I was actually, talking and lucky to have somebody at Duke University that came up with a very smart, very intuitive way of selecting the number of factors in a factor analysis model. And what they do throughout the Gibbs iteration, throughout the inference of the factor analysis, they look and they ask the question, is the current number of factors optimal for our analysis? Are there any loadings that are zero across all factors? If that's the case, you might want to reduce the number of factors, and you dynamically adapt that and you run your analysis. And what I'm showing here as an example is for stage range 7, 8, you can see how throughout my analysis of my factor analysis model, the number of factors actually converges to roughly 60 factors, whereas the later stage 11, 12 goes somewhere in the area of 140. As expected, the later you go in Drosophila embryogenesis, you would expect to use more factors because more biological processes are taking place. And so at this point, we had our features. We had these sparse factor analysis features extracted from our images with a number of features dependent on your stage range. And the next question we had in mind was how are we going to deal with transitions with temporal dynamics. As I mentioned in my first or second slide, you can clearly see some development for any given gene with time. So you would really like to look at these transitions. In the previous work that we published in 2011, we used the sparse Bayesian factor analysis features, and we used the SVM classifier on top of those features to do gene annotation. So what we did, we built SVM classifiers for every single stage and every single annotation term, disregarding any transitions across these stages. And we did show that the results in terms of accuracy were as good as state-of-the-art models with bottom-up approaches, but now we really wanted to do better than that. We still wanted to use the same sparse features, but on top of that, we wanted to use some transitional information that we had access to. So we came up with this conditional random field model, which is different from an ordinary classifier like an SVM in a way that predicts a label regarding the neighboring samples, and which, in other words, it takes context into account. And I'm going to show you in the next slide like the graphical model for such a conditional random field. I showed here a picture that really represents our model. So we have 5 different stage ranges for Drosophila embryogenesis, and every single column represents one stage range. And for a given stage range, I'm showing you the annotation terms that we selected, the most used ones. And we had 14 annotation terms in stage range 4, 6, all the way to 8 annotation terms in stage range 13, 16. So the question would be simple. If you just built an SVM classifier and you like to annotate this annotation term for the given gene, you'd like to answer the question, should this node be a 0 or a 1? Should this annotation term annotate that gene or not? And you will move to the next one and so forth. But you'll end up building 48, in this case, 48 SVM classifiers, which is time consuming, and it's just computationally expensive. And so using a CRF, you would actually be able to use transitions across stage ranges and within stage ranges on top of these potentials for every single node, and with the hope that we would actually improve our annotation accuracy. Just give you more details. The inference we used was a junction tree inference. We can, of course, talk offline if you have any more questions about this model. The node potentials for every single node in this framework was represented by probability densities of multivariate Gaussian distributions on the sparse vector loadings. So similar to an SVM, you would train your SVM on your sparse vector loadings. Here we use the same factor loadings, but then we computed densities of multivariate Gaussian distributions. So these were the node potentials, and then the edge potentials were rather simply computed by doing counts. For example, I would ask the question, how many genes are being annotated by this annotation term, and how many of genes are being annotated by this new annotation term here, and they'll just go and do a ratio proportion of the entire number of genes, and this is how we'll get all these blue and red annotations. Now, clearly we're expecting to improve using this conditional random field. We're expected to improve results over standard SVM, independent SVMs for every single node. But then we also realized that there were a few interesting scenarios that came in place. For example, what if we only use transitions within stage ranges? So we're going to ignore all the transitions that happen with time. We're going to only look at these red line transitions, and we're going to run some numbers to this, and I'm going to show you results later. The next scenario was, what if we only take into account transitions across stage ranges? What would happen? Would it still improve our results or not? And ignoring transitions within a stage range, and I'm going to show you results for that. And the third scenario would be, what if we actually take into account all the transitions that we have access to, hoping that this model would do improve over standard SVMs. And results are here, very simple and very clear. We had, in order to compare to a standard SVM framework, we averaged across all annotation terms and across all time points. We averaged an annotation accuracy, and we obtained this 83.32%, which was already above competition, given the fact that we already used here sparse factor analysis features, and we already proven in the paper in 2011 our advantages. But now we wanted to show even more. We wanted to show what would a conditional random field do to our framework. And the first scenario is rather maybe surprising a little bit. It's rather close to a standard SVM. So within stage transitions didn't really help in terms of annotation accuracy. However, when we move to the next scenario using temporal, using this type of transitions, we did show an improvement, whereas the third scenario, which would actually use all kind of transitions, that will show an improvement of 3.4, 3.5%. Now you may say that this is not such a significant improvement. However, I'd like to remind you that we're already doing quite well here. We used all the entire set of images that we had. We didn't do any manual selection over images as other people or other papers would do. We just wanted to see how reliable our model is. So given that so many images would be out of focus or would be different orientations of the embryo, we never really expected to do in the 95, 98% area. And on top of that, just this increase is we did find it very, very significant, because to get this accuracy we had to use the ground truth, which ground truth was provided by the professional curator at Berkeley. And of course we actually showed in this paper that there were a couple of scenarios where we believe that the curator was actually wrong annotating a given gene, and that could also might be a case of lowering your final annotation accuracy. And the last thing that I pointed out at the very beginning was how to deal with missing data scenarios. And luckily, a CRF is able to deal with such a scenario, because let's suppose that in the same framework we have here, we just do not have any image from stage range 910. Well, a CRF, what would do, would still be able to reconstruct, would still be able to label all these nodes, the remaining nodes, given that something is missing, just by integrating this part out. In other words, we'll go from this node to that node by summing across all the transitions that are just not present. And so with this in mind, we're actually very excited to see how the CRF would perform in a missing data scenario. So The results shown here on top are the full data set, the 86.7% accuracy that I showed you at the beginning. And then I'm showing you what would happen if we would manually remove randomly 5% of the data. And clearly, interestingly enough, if we just remove 5% of our data, we are not really doing that much worse. And even more interestingly, if we remove up to 15% of our data, we are still doing better than independent SVMs on every single annotation node, which I think it's quite remarkable. And I'm also showing here some examples in which, for this very gene, I removed this image from a stage range 4-6, and we're able to annotate accurately the next 4 stages with an accuracy of 78%, or even lower. If we remove the stage range 13-16, we're still able to reconstitute the entire set of four labels for the previous stages. And so, to wrap up, we did talk about dealing with massive and complex data sets, and I show you this interesting factor analysis model to deal with the high-dimensional data with a smart way of extracting features. I also dealt with the time-dependent modeling, which we all have if we deal with maybe Drosophila embryos or C. elegans or even cancer progression data, and also missing data analysis. And I would suggest if you are interested in studying heterogeneity or even local homogeneity in any high-dimensional spaces, it's probably a good idea to look into non-parametric Bayesian models, as a way to find flexible models. And with that, I would like to thank all our lab, my PI, as well as William Majoros, who is right here and is actually also part of on the paper, David and Neil. Also, Erwin Friese, who was the guy in charge of collecting all these images, and he was really helpful in giving us new annotations and whatnot. And I'd also like to thank to ISMB and ECCB for generously providing me with a travel fellowship. Thank you.
Yeah, again, time for questions. There was one.
Yeah, so a nice talk. It's very interesting work
Thank
as well. I'm wondering if you could say something about how often within a stage you see multiple annotations. I mean, that might be a reason why you're not getting very much information out of the transitions, is that you don't get multiple annotations within a stage.
You mean for a given image to have multiple annotations?
Yeah, that's right. Yeah.
So that happens, right? So let me go back to like for this case, it's quite often that for a given stage range and a given gene, you're going to have more than one of these nodes to be a 1, right? So you have multiple annotations annotating that given gene for a given stage range. So it's not always the case that only one annotation term is annotating a gene. I'm not sure if that's your question or.
Well, the question is trying to get at why you're not getting any information about modeling the covariation of
right.
labels within a stage. It seems like you should get some information out of that. But
yeah.
If you have very sparse annotations.
I understand. Well. You're right. So you mean that the red transitions, right? They didn't really improve too much. Yeah, I'd probably like to think more, of course, about it. And yeah, we're surprised. We really expected to see an improvement even within stages. But I think I'll probably have to go back and really look at the transitions, see the model, and see maybe there are some genes or some patterns that really hurt the model. So overall, the accuracy doesn't really get too good. But it's an interesting point, of course. Yeah, so yeah, so the feature extraction method, right? It's quite fast. If I remember correctly, what we published, we said we had about 2,000 genes that would be around 10,000 images, and to extract features will take within 2 or 3 hours. So, and that's usually do this offline anyway. So it was really not a concern. So within a couple hours, and we wrote this in MATLAB. Of course, you can speed it up in C or Java or some other things, but that was quite decent. Could you repeat, please?
Sorry? Is it right that every feature is a pixel in an image?
No, so that's a good question. Let me go back to, I didn't really have time to go through every single detail. Maybe here. Huh? All right, so original images will have a resolution of, let's say, 100 by 200 pixels. And so if you just represent it as a vector, you end up with two 2 million pixels, right? But we didn't want to go to that big space, so we used a fixed grid. We pretty much downsampled. We used like a neighborhood of 9 by 9 pixels, and we computed an average for those 9 by 9 pixels and move on to the next one. In a way that we went from a 100 by 200 pixels to a 40 by 80 pixel image, and we converted that into a 3200 dimension vector.
Okay, but so every feature is a 9 by 9 area.
Yes. Yes. Well, so let's make that clear. That's in space X, in my original data. Later on, if you talk about the Bayesian features, that's different. These are the features estimated throughout the model. So they don't really represent one given fixed area within my data. These are the estimated features later on. All right.
Have you ever contemplated hierarchical feature model where you start off with like high-level classification, a broad general classification, and then within the class, within the features use like sub-features and so on and so on.
So are you talking also still about a sort of a top-bottom approach? So you look at all the features and you hierarchically go down and cluster those? Do you have a model in mind? Just so I know exactly what, like, give me an example of that. Because there are, I want to understand your question better than.
Yeah, like, I mean, imagine, you have intensity in the head and intensity in the tail, or in the body. And then within intensity in the body, we have basically, sub-features like intensity in the chest and intensity
Right.
in the belly.
So maybe that, right. So maybe in my mind it would come that maybe wavelets would do something like that. Or we could definitely improve our features if we use some spatial information. We did not care of any spatial information in our images, and of course that is something that if somebody is interested could totally apply to that. Yep. Sure.
Can you still comment a little bit on the CRF part? I mean, about the feature functions and also
Sure.
and also about your implementations. CRFs were not particularly well known for people
Right, so-
past 10 years ago. So are you using your own implementation?
Well, so we had to. It was not really my own implementation. There are quite a few even available nowadays. We had to adjust some of my implementation in terms of time because we ran into issues of computation. There were cases where we would even finish in like a few days. So there were things in terms of distribution, in terms of computing the node potentials and edge potentials. Of course, that's a situation. But definitely in terms of time consumption, I would say that. The CRF was definitely the lengthy one of all my work. Would take just to run across a set of, let's say 1,000 genes would take, I don't know, a few hours, but like maybe 6-7 hours or so. So that's quite lengthy. We did try to, and we would run sometimes into issues of, space allocation and all that. But I think there is room for improvement into that. Absolutely.
Perhaps one more question.
One short question.
Well, I would have one.
So you have a question, sure.
Yes, you did one run of cross-validation, is that right?
Tenfold cross-validation, yes. Yeah.
Tenfold cross-validation.
Yeah.
Because, I mean, if you have lots of, annotation terms, then it's virtually impossible to do something like stratification. So if you had one run, then I presume there would be a lot of variation per term. Right.
Well, I actually have to go back. I'm pretty sure I did. We used tenfold cross-validation, but I think I did 5 different runs across this tenfold. It's just we did play a lot, and even we had the results from the factor analysis part, and we had an idea of how stable and how. Yeah, there was definitely we had that into consideration. Absolutely.
Okay, then